Let's take a look at the second step in urine formation, reabsorption. Reabsorption is the movement of some of the water and solutes that got filtered into the nephron from the nephron back into the blood. Okay, so reabsorption is taking substances that entered the kidney and then putting them back into the blood. And so let's just take a look at what the um, glomerular filtrate consists of. And so taking a look at this list, the glomerular filtrate is kind of like a subset of plasma, right? It contains water, it contains electrolytes, some small nutrients, small enough to pass the filter, glucose, amino acids, okay? And so we have nutrients, and then we have waste products. And so you can see what gets filtered is not just waste, it's just a separation of large from small, remember? And so what gets filtered is water. We can definitely use that. I would want to take that back into the body. Same thing with electrolytes. We could take some of those back and use them, right? Nutrients, why get rid of these? Let's take that back. And that's what reabsorption is all about. Reabsorption is about taking these small solutes and water from the filtrate and putting it back into the blood. And at the same time, we don't want to take back the waste products. We'll let that sit in the renal tubule and become part of the urine. So with reabsorption, most of it occurs in the proximal convoluted tubule, and the cells of the proximal convoluted tubule even contain microvilli, which increase the surface area for reabsorption. So most reabsorption occurs before the filtrate even hits the loop of Henle. So taking a look at a picture from our book, what they're showing us in this picture is in yellow. This is what the filtrate looks like. Okay, so we're looking inside of the proximal convoluted tubule here, and then they're showing me the cellular wall of the proximal convoluted tubule. Okay, the interstitial fluid, and then here's the blood. So I know that reabsorption is basically taking items from our filtrate and putting them back into the blood. And so they show us a lot of detail in this picture, and we're not going to be concerned with all of this detail. No worries about that. But I just want to show you that the red arrows are indicating active transport. The blue indicate passive transport. And so I just want to take a look at that for a second. So do you notice that with the red arrows, sodium, glucose, amino acids, that some things are going to be reabsorbed using active transport, while other things like water, some ions, lipid-soluble substances, some other substances will be reabsorbed passively. And so there's going to be a combination of active and passive transport. So let's take a look at the active reabsorption of some of the nutrients that are in the filtrate first. Let's take a look at glucose. And so to look at glucose, I'm using a Netter illustration from Elsevier, and what they're showing us in this picture is the afferent arteriole bringing blood into the nephron. In the afferent arteriole, there are glucose molecules, the black dots, and you can see that 100% of the glucose is being filtered. And so when I look at the efferent arteriole, I don't see any black dots in there, which are representing glucose. So glucose comes in, 100% of the glucose gets filtered, and so there's all the glucose in the proximal convoluted tubule. What they do in this diagram is they're using this kind of like turning wheel as an analogy for active transport. Active transport uses a carrier, right? It's carrier-mediated transport, which means that the carriers, you only have so many and they can only work so fast. And so they're just showing this turning wheel analogy, showing that glucose is moving on to, get actively transported, and it's getting kicked back into the blood of the proximal convoluted tubule. So that looks good. Let's take a look at another picture. In the next picture, what they're doing is they're increasing the blood glucose concentration. And so first picture, I see that, normal blood glucose. This is also a normal blood glucose concentration, but say like towards the upper end of the range. And so there's more glucose in the blood. Still, 100% of it gets filtered, and 100% of it should be reabsorbed. Okay, we should have enough carriers, and those carriers should be able to kick back all of the glucose from the filtrate into the blood, and we can see that happening. But you can also, when you make a comparison between these two pictures, hopefully notice that in the diagram where we have a lower blood glucose concentration, do you see how not all the carriers, not all those slots on the turning wheel are in use? Whereas when the glucose concentration is a little higher, I can still reabsorb everything back into the blood. So in other words, once you pass this carrier, there's no more glucose in the filtrate, in the nephron, right? And all the glucose is getting kicked back into the blood, but I'm carrying that glucose back in at maximum capacity. So let's take a look at what would happen if our blood glucose level went above the normal range. So taking a look at this third picture, what they're showing us is a very high blood glucose level. This would be above the normal range, which is not what we would expect, right? That's abnormal. That means that insulin and glucagon are not working to keep our blood glucose within the normal range where it should be. But other than that, 100% of the glucose gets filtered, so all that glucose is entering into Bowman's capsule, into the proximal convoluted tubule. The carrier is working as fast as it can, right? We only have so many carriers and they only work so fast, and it's kicking the glucose back into the blood. But unlike the other two pictures, take a look at this. Now that capacity of the carrier is not enough. There's so much glucose in the blood that now some of it is missing, getting reabsorbed, and that is the glucose that's showing up in the urine, and so that would be considered abnormal. As long as our blood glucose level is kept within a normal range, then we have the ability to reabsorb 100% of the glucose into the proximal convoluted tubule. Once that glucose is no longer regulated by hormones, it's above the normal range, then we can no longer reabsorb all of it, and some of it shows up in the urine. So this picture is about glucose reabsorption, but I just want to take a quick look at this picture and think about amino acid reabsorption. We were saying that amino acids are also going to be actively reabsorbed back into the blood in the proximal convoluted tubule. And so if you think about amino acid concentration, think about this. Do we regulate the amount of amino acid in our blood? And the answer is no, we don't regulate the amount of amino acid in our blood. That would be just a function of our dietary intake, right? So if we drank a protein shake, then this would be higher compared to if we just had carbs. And so because we don't regulate the amount of amino acid, even though amino acids will get filtered and amino acids will be actively transported, reabsorbed, I should say, back into the blood, you could have some amino acids spilling over into the urine, and it would be okay in that case, just because we don't regulate amino acid concentration. So you can see amino acids spill over into the urine. We should not see glucose spilling over into the urine. Taking another look at our picture of reabsorption, we can see that sodium, we spend energy to reabsorb sodium back into the blood, and when you reabsorb sodium, that's going to make the blood more positively charged, which will attract negatively charged ions passively, and it will also make the blood more concentrated, and so that's going to attract water passively by osmosis. So to summarize reabsorption, what we're saying is that most reabsorption occurs in the proximal convoluted tubule. That reabsorption means that we're going to have substances taken from the proximal convoluted tubule back into the blood, probably of the peritubular capillary. And we're saying that glucose, as long as our glucose is being regulated properly, then 100% of the glucose that entered into the filtrate should be reabsorbed into the blood actively. That amino acids, depending on our intake, some might remain in the tubule and become part of the urine, and that's okay, but we're going to actively reabsorb amino acids. Sodium, we use active transport, and so we're going to actively take back about 70% of the sodium that's in the filtrate. So most of the sodium that we intake, we're going to actively take that up into the blood. And what we were saying is that when you spend energy to bring sodium into the blood, is that because sodium has a positive charge, you're going to create an electrical gradient. And that electrical gradient is then going to passively attract negatively charged ions. So there are negatively charged ions in the filtrate. I'll just, give one as an example. And I'm using blue to show passive transport. And so negatively charged ions will passively follow. And I'm going to draw enough negative charge in the blood to show that they would follow to neutralize that electrical gradient. So we spend energy to get the sodium. Negatively charged ions follow. Blood becomes very concentrated. And so what will happen is it becomes more dilute in the tubule. And so water will move just by osmosis, which is the passive movement, right? Water will move from its area of higher concentration, the dilute solution, into the area of lower concentration. And so water will be taken up passively. And this is just a summary of reabsorption within the proximal convoluted tubule.